alumni, Dr. Arun Kumar. He's currently working as Associate Professor at CMC Vellore. He did his MD of the batch 2012, and then followed by which he did his postdoctoral fellowship in molecular hematology from CMC Vellore. He has continued his studies and got trained from Cincinnati, Ohio, and College of Wisconsin, and he has been receiving several prizes and attending several conferences as faculty in addition to his various papers. The recent one to his credit is CMC Senatus Prize for the year 2021 for his work on molecular diagnosis and immunodeficiencies. So I'd like to present before you Arun.
Thank you, ma'am, for the introduction, and thank you, sir, Millie ma'am and Rashmi ma'am for arranging this. Feels really good to be standing here again, and lots of nostalgic memories. Not very long ago, I was on the other side. I was sitting over there as a student, 2012 till 2015, in fact. So it's good to be back here and step again inside this beautiful department. So what I will be doing over the next 40-45 minutes is to just give you some idea on various molecular techniques available for more so from a hematological perspective, but the technique remains the same whether you're looking at solid tumors or hematological malignancies. The idea being, so when I was a student, I thought I was very interested in hematology right from the beginning, and I thought the whole world in hematology is morphology. But the moment I stepped out, I entered into CMC, I realized that there is so much more to learn, so much more to happens, not just morphology, there are many other things to know. And just to give you one example, like I said, I was all into morphology, and the exam that I gave, my ND exam in 2015, officially that was the last time I saw a morphology slide or I reported any morphology, because right from then, it has all been molecular techniques, flow cytometry, and all the other ancillary techniques. So there is so much, it may not be still, it's not accessible in every part of the country for sure. But, now it's a lot more available as compared to when I was a student, and you're all beginning your careers right now. So, you have another solid 30-40 years down the line. So it's very important that you get little bit oriented to all these molecular techniques, even if you're not practicing it in a regular basis. With that background, I will just, so like I said, my talk will largely focus on hematology perspective. So we have come a long way from the era of morphology, cytochemistry, and immunohistochemistry. Slowly, one by one, all the techniques came about. Starting with karyotyping, which helped us to classify hematological malignancies to some extent. A major breakthrough was flow cytometry, which really simplified the diagnosis of all hematological malignancies, at least the leukemia ones. And then you had FISH, and the next major breakthrough was the invention of PCR, and that really changed the landscape of how we look at not just hematological disorders, any disorders, for example. We started exploring for the reasons why something is happening and not just trying to diagnose the entity per se. And currently we are in the era of next generation sequencing. So just for you to get an idea how much molecular diagnostics and all these techniques actually make a difference. Say this was, like I said, if you're thinking morphology is everything, say you get a slide like this, it's quite evident. You have nice OR rods and clear-cut blast. It may not be difficult for you to say it's a case of AML. But this doesn't happen always. You have a lot of the times your morphology might be a little confusing, and no matter how good a morphologist you are, nobody can say I'm 100%. I see 100 cases, all the 100 cases, my diagnosis is going to be right. It will be wrong on a few occasions. So if you're restricting yourself to morphology, all you can say is probably this one is quite evident, but some cases you will say probably it's a case of acute myeloid leukemia. You will get a little bit more clearer picture when you do, cytokine. You look, apply SBB or MPO, and if it get it positive, then probably you can say with little more surety that it's a case of AML. So moving ahead, if you had access to flow cytometry, markers like 34, 117, 13, 33, HLA-DR, they all come positive. Now you can be very sure. You can say with 100% surety that it's a case of AML. There's no doubt in my diagnosis. But that's all you can say. All you can say is you can give an accurate diagnosis. You cannot tell the clinician, how this patient is going to behave well, he's going to behave very poorly, how is he going to respond to the treatment. All that are still in question. Move further ahead. If you have access to cytogenetics, this patient, let's say he had translocation 821, the RUNX1, RUNX21 translocation. So is this good or bad? Yeah, so RUNX21, 821, is a good prognosis. So probably you can say, this patient will be having a good prognosis and, outcome may not be very poor in this case. So this is if you have access to cytogenetics, either karyotype or FISH, or to some extent even RT-PCR. Suppose you also have access to molecular techniques. The same patient, let's say, it's an AML, so obviously you will look for NPM1 mutation, FLT3 mutation. If the same patient has NPM1 and FLT3 mutation, then now you are in a soup. You thought it's good prognosis, but co-occurrence of NPM1 and FLT3, now maybe you'll say probably not as good as we thought it would be. Probably it will fit into an intermediate prognosis. Okay. Going one step further, so till few years back we were till at this level. Now we have access to NGS also. The same patient, when we do NGS, we identify say TP53 and ASXL1 mutation. So now you see that it's completely changed. When you get TP53, it overrides anything and everything else that you have found. Now it is definitely a poor prognosis, a very poor prognosis. So you can see how much it is changing from a time when we were giving a probable diagnosis. What we thought was a good prognosis initially is now changed to intermediate and further changed to, a poor prognostic indication. So this is an hypothetical scenario. This is how much difference, you can have based on, if you have full access to all the molecular techniques. And if you have, we also do flow cytometry based minimal residual disease tracking, you can also tell, at the end of induction whether it needs to be intensified with the consultation has to be done with the transplant or we can continue with chemotherapy. So there's so much information which is required, not just giving a diagnosis. So clearly, cytogenomics is important. Some scenarios, this was an example with leukemia. There are scenarios where the diagnosis itself will be reliant on having a molecular, identifying a molecular aberration in that patient. So clearly cytogenomics is important. So for you to understand, basically any molecular test that we say, there are just two categories. It's a test that starts with DNA as the starting material. There are tests that will start with RNA as the starting material. Now, why this difference? It depends on what you're trying to look at. Usually, if you're trying to look at gene expression, these may sound fancy terms, but believe me, they're all simple. So if you're looking at gene expression, or say, for example, you're trying to pick up fusion transcripts by RT-PCR or RT-PCR, we start with RNA, then change it to complementary DNA, and then we go ahead with a PCR. For most of the mutations that we look for, it is the DNA that is the starting material. And depending on what we are trying to identify, the type of molecular test that we do will vary. Okay. So we talk about mutations, you should have some idea what these mutations are. Okay? In fact, as far as leukemia, not leukemia, any malignancy is concerned, mutation is actually not a relevant term anymore. We call it single nucleotide variants because these are not monogenic disorders. So it's not, one change which is going to result in leukemia. It's a sequence of mutations acquired over a period of time that may result in the phenotype. So the type of mutation will determine what molecular test you will be doing. So if it's a change happening at the level of chromosomes, okay? So you know the hierarchy, right? You have chromosomes. Within the chromosomes, you have the DNA, and in the DNA, you have genes, and in the genes, you have the exons and the introns. So if you're looking at changes which are happening at the level of chromosomes, we do it by, say, karyotype or FISH or RT-PCR, etc., where we are trying to identify huge changes which is happening for so many kilobases of genetic material. If the change is restricted to a small part of the chromosome, deletion running for a few thousand or maybe 10, 20,000 bases. So it's relatively a shorter change compared to a major, say, trisomy 8 or a monosomy 7, something like that. So this is in a very small portion of that chromosome. So this we cannot detect by karyotype. So this we will do by various techniques like NGS, or we have something called as MLPA. I'll maybe talk about it later. Now, even within this, so this is extending segment of DNA. So within this, if you are looking at changes happening at the level of genes, one single gene, or even within a single gene, if it's only 1 or 2 exons which is getting deleted, now it's at a much smaller level. Okay? So again, the type of test that we do will be different from the previous scenarios. Here we will do something called as a deletion custom-made PCRs, or again MLPA can be used. If you go down even further, within the exon, if you are looking at changes happening at the level of single nucleotide bases, okay? You know what the nucleotide bases are: adenine, guanine, thymine, and cytosine. So if it's a single nucleotide variation, meaning at one particular position, say, so the black ones are G, guanine, and the red is T. So in this particular position, you're supposed to have only black, but you're having one black and one red. That means in one allele, you're having guanine, in the other allele, what is supposed to be guanine is now mutated into a thymine. Okay? So this is called as a single nucleotide variation, and this is what we call as point mutations. And this can be detected by techniques like sequencing or even NGS for that matter. So depending upon the size of the change that is happening, it comes down all the way from a karyotype down to single nucleotide variations picked up by sequencing. Okay? So how do we do molecular diagnosis? In a very broad sense, there are just 3 or 4 basic steps. So you need all these tests, like I said, happens with nucleic acids, DNA or RNA. So the first step will be to extract these nucleic acids. So where do we have if you take blood, for example, where do you get these DNAs? Yeah, so DNA should be present in the nucleus, obviously. So any nucleated cells will be having this DNA. Okay? So if you take blood, your platelets are enucleate, your RBCs are enucleate. What you're left with is the WBCs. So WBC is our starting material. So it is just logic. If you take peripheral blood, so if you have to get the DNA, you have to go step by step. So first step is you need to remove all the RBCs and platelets. So you do an RBC lysis, so that you are left. If you do a spin, you'll be left with the WBC pellet. The next step is to bring out the DNA from inside the WBCs. So what do? You open up the cell membrane, and the next step is you open up the nuclear membrane so that the DNA comes out. So, but once you open the cell membrane, what happens? Along with whatever is there in the cell, all your proteins, all your cytoplasmic RNAs, everything will also come out. So the last step is to separate the DNA from all these other constituents. So once you separate and get the pure DNA, that is your starting material. So you take this, do a PCR. Any molecular test starts with a PCR. Okay? So you do a PCR, and then at the end of PCR, if depending on what additional, if you're looking at deletions or etc., the PCR might alone suffice. But sometimes you will do additional techniques like sequencing, etc. Okay? So different ways by which we can extract the DNA, not going into all the details. But the concept is like what I said just now. You lyse the RBCs, then open up the WBCs and take out the DNA, and then purify the DNA, removing all the non-DNA particles there. If it's RNA, the last step alone will vary. You have RNA on one side. For RNA extraction, your DNA will be considered an impurity. Likewise, in a DNA extraction, your RNA is a contaminant. So you just have to separate them out and then have the pure DNA or RNA, whatever you're looking at. So we have manual and so we were doing it manually. Now we have various equipments to do extraction automatically with all those robotic arms which will directly extract and give you the DNA. So once you have extracted, your whole molecular assay depends on the quality of the DNA you have extracted. So we have to assess the quality, which can be done again by multiple methods. This is one set method which is commonly used, a spectrophotometer. So without going into the details, it depends on the shape of. If you get a nice bell-shaped curve like this, it means your DNA quality is good. Whereas if it's a degraded DNA or if there are a lot of contaminants, you get something like this, a very haphazard, looking graph over here. So once you have a pure starting material, the DNA, you can go ahead with your molecular tests. So again, different ways by which we assess the quality and quantity. So the next step is a PCR. Now it's okay if you don't understand any of the molecular techniques that I talk about, but it's very important that you understand PCR in detail. So the basic concept of PCR, I hope you're all aware of it. In a nutshell, your PCR, you have your starting material, the DNA that you have extracted. What you do is you add whatever reagents are required for the PCR to happen. So what do you need? You need all the nucleotide bases, and we call it as DNTPs. Okay, and then nothing but the adenine, guanine, thymine, and cytosine. And then you need the primers. So the primers are nothing but that will initiate your amplification. Okay, so and essentially you will have two primers, one forward and the reverse primer. Why? Because your DNA is double-stranded. So once you denature, you have two single strands running in opposite direction. One in 5' to 3', the other way around. But so when you have two primers, one will, so these two strands, you can call it as left strand, right strand, or strand 1, strand 2, either ways. One primer will amplify the first strand, the other primer will amplify the second strand. And this will happen n number of times, ultimately giving you billions of copies from a single DNA template. Okay, this is basically the PCR. You have 3 steps, like denaturation, annealing, and extension. Okay, so your PCR, even if you take RNA as a starting material, first step is you convert it into cDNA, okay, and then you take it to the PCR. So why can't we use an RNA in a PCR? Okay, the answer is because what is the enzyme that is required in a PCR? Yeah, what is that polymerase? Yeah, it's a DNA. So for the PCR to happen, as you can see over here, your PCR has multiple 3 steps basically happening at different temperatures. At 94 degree is the denaturation. Then you cool it down to 55, depending on the primer sequence. It can be 55 to 60 degree, and then your extension happens at 72 degree. So you need a polymerase enzyme which is heat stable. And the only heat stable, because that should be stable even at 90 plus temperature and also at 55 degree. So the only heat stable polymerase that we have is a DNA polymerase. Your RNA polymerase is extremely labile. Even if you change the temperature by 1 or 2 degrees, it starts degrading. So that is why even if you have to do a test based on RNA, we convert it into cDNA, and then because it's DNA now, we can use the same DNA polymerase and do the PCR. Okay, and you get the 2 power n times amplification with each and every cycle, and then you have. So what will happen now is you have extracted is the DNA. So your DNA has nothing but all the genes, but you're looking to identify. You'd say you are looking at the case of thalassemia. You want to identify mutations in the beta globin gene. So from the DNA that you have extracted, you need to selectively amplify only the beta globin gene, so that then you can do whatever downstream assays in that. So to selectively amplify the beta globin gene, you will design primers such that one primer, say if you're covering exon 1 of beta globin gene, you will have one primer before the exon 1, the other primer after the exon 1, so that the whole exon 1 is covered from either side. Okay? This is how basically your PCR works and how we design primers for the PCRs. And do stop me in between if something is, you're not able to follow something. And then at the end of PCR, what we do is we do a electrophoresis to make sure that your PCR amplification has happened properly. If it is good, you get a nice sharp single band like this. If for some reason either the DNA was not good or your PCR you have contaminated during the setting up the PCR, instead of getting a sharp band, you get a more hazy picture over here. So if it's say something like a deletion PCR, with the presence or absence of a band itself, you can confirm that the mutation you are looking for is there or not. Sometimes we need to take this PCR product and do subsequent additional assays. Like, for example, if you're looking at point mutation, the same example I gave, a beta globin gene, the next step we do is we sequence the beta globin gene from this PCR product, and then we read the sequence and see if there is any single nucleotide variation. Like here you can see what is supposed to be T in a normal individual has become G and it's homozygous, meaning there is no So heterozygous, homozygous, meaning if you have one normal peak and one abnormal peak, you call it heterozygous. If both, there is no normal and you get only one abnormal peak, it is homozygous. Okay. Or there are other assays that we can do subsequently. So I'll just give you a few scenarios, and in those scenarios, we'll talk about different molecular assays. One is thalassemia, few on the benign side and few on the malignant side. Okay, so usually the first question will be, do we need molecular diagnosis in, say, for example, a thalassemia? How many of you say yes? We need to have a molecular diagnosis to diagnose thalassemia, or you can say I can confidently diagnose with my peripheral blood and, maybe not peripheral blood, but with an HPLC for sure. So yeah, you may not need in all scenarios. Your HPLC is, it's an extremely useful test, and most of the times it can give you a clearer picture. But every diagnosis that you make by HPLC, you will have at least 3 or 4 differential diagnoses. If you understand HPLC in greater detail, you will know that there are various parameters that can modify the result. Even the same picture, say for example, if you have a sickle with 80% and 20% HbF, the same values could be a homozygous sickle, same values could be a sickle beta thalassemia also. So one easier way will be to just take the parent sample, do the same HPLC in the parent, that will confirm the picture. But if that is not available, then it's a better option to do a molecular diagnosis to confirm whether it's homozygous sickle or a sickle beta thal. Other reasons why we do it is you get a good phenotype-genotype correlation, and it helps because there are some mutations which causes thalassemia major, some mutations which cause intermediate, or what we call as beta 0 mutations or beta plus mutations, depending on the severity. So all this information you can get if you do a molecular diagnosis. And of course, the most important reason for any germline disorder to do a molecular diagnosis is not for the patient, but for the subsequent pregnancies, okay? The prenatal diagnosis and genetic counseling. So when I say germline somatic, anything which is inherited, that is what we call as a germline mutation. Okay? And germline mutation is either from the father or from the mother or from both. So it is always either heterozygous or homozygous or normal. Whereas somatic mutation, we don't talk about heterozygous or homozygous because it depends on what percentage of cells are carrying the mutation. Okay? It is still hetero-homozygous, but you cannot label it because one patient may have 95% blast, the other patient may have 20% blast. So the percentage of that peak that you see will not be the same in 2 patients, even if both are having a homozygous mutation. So there we just call it a somatic variant. In germline, we call it heterozygous or homozygous. Okay? So just to give you a brief idea on how to approach a case of thalassemia, probably here you are doing till this level, till the HPLC. So yeah, it always starts with history and CBC for sure. And then you do a HPLC, which 9 out of 10 cases will give you a correct diagnosis if you are doing the HPLC for the patient and the parents. So if you want to go further, then we go for the molecular diagnosis. So that will depend on whether you're looking at a case of beta thal or an alpha thal. So in beta, generally we have point mutations, whereas in alpha thalassemia, it is deletions. Okay, this you have to know. We have the point mutation that cause beta thalassemia is specific in specific populations. Okay, there are some 8 or 9 mutations which are probably seen in 90% of the Indian thalassemia patients. So we first look for those 8 common mutations with a very cheaper, assay. Like I said, 9 out of 10 thal patients will have mutations in one of these 8 mutations. If that is negative, then we go for sequencing the whole beta globin gene. Sometimes it could be a case of, what we thought as a case of thalassemia might be a delta beta thal, or it could be an HbFH. These are all deletions. So if the point mutations are negative, then we go for identifying the deletions by using a technique called MLP. Okay? If it's alpha thalassemia, we proceed the other way around. We initially look for the deletions because, as you all know, I'll show that in the next slide, whether it's a silent carrier or a alpha thal carrier or HPH disease, we generally call them based on the number of alpha genes deleted. Single gene, 2 gene, 3 gene deletion. 4 gene, of course, the child will not be born, it will be high drops. But so we start with looking for the deletions. Again, different methods are there for that. We commonly use MLP now. If the deletions are negative, you have to remember that not all alpha thalassemia cases are caused by deletions. There are some point mutations also which can cause alpha thalassemia. So if deletions are negative, we look for point mutations. Okay. So this is the, we do something called as a reverse dot plot assay, quite similar to, say, for example, IHC, based on a hybridization technique. Just like IHC here, what you have is the top row will represent, we'll have probes that will identify the normal sequences. The bottom row will have probes attached to it, which will identify the mutated sequences. So when you do the PCR and then subsequently add this membrane, if you get a dot on the top row, then you have a normal sequence for that particular, codon position. If you get a dot on the bottom row, you have a mutation. It's as simple as that. I'm not going to the technique, but the basic concept is it helps you to identify whether it's a normal sequence at a particular position. So these are the common mutations in our population. So it will tell you whether at that point it is normal or there is that particular mutation. So this is one easier or rather cheaper way of identifying the mutation. So here are some examples. So here you can see this patient has one dot in the normal also, one dot in the mutation also. So that means in one allele it is normal, in one allele it is mutated. So this is what we call as a heterozygous mutation. Okay. Whereas here if you see, here for example, this is this particular column is for sickle mutation. So you can see there is no dot here and only a dot in the mutation sequence. So this is a homozygous sickle mutation. Okay. So this is how we call something as heterozygous or homozygous. So if that is negative, we do sequencing. So if rarely, if say it's a patient coming from Punjab or the Sindh, province, there is one particular deletion which is quite common. So we test for that particular deletion using a PCR. But otherwise, generally we go for Sanger sequencing to pick up. If the RDB is negative, we do the Sanger sequencing. Okay. So yeah, I'm not going to the techniques and all. Maybe if you're interested, you can read about it. The idea is not to make you experts in the different molecular assays, just to tell that these are the different assays that are available, and maybe you can just get an idea what all is done for what particular diseases. So if there are no point mutations, and if you're looking at deletions, so this is an example. So the basic concept is so if it's what we do is we make a ratio between a normal and the patient sample. So if the patient is also normal, that means normal by patient will give you a ratio of 1. The patient has a deletion, the ratio will be 0 or 0.5, depending on whether it's homozygous deletion or a heterozygous deletion. So it's as simple as that. So here if you see, this one, the ratio, it's all coming close to 1. So that is a normal individual. Whereas here if you see, this particular stretch of dots, they are 0. The ratio is 0. That means there is a deletion starting from this position to this position. Okay? And it's a homozygous deletion. Whereas here if you see, the same stretch as you saw in the previous image, but here you are getting a ratio of 0.5. So what does this mean? It's a heterozygous deletion. Okay, so this is how we identify whether it's a heterozygous or a homozygous deletion. This technique is called MLPA, multiplex ligation probe amplification. It's okay if you're not able to understand the technique, but basic idea is compare it with the normal and say whether it is same as the normal or you are having some loss in particular regions, which will tell you that there is a deletion. So that was beta. If it's alpha thal, like I said, we start with looking for the deletions because they are far more common. And so when we say alpha deletions, we say so we generally talk about single gene deletion, 2 gene deletion, 3 gene deletion, which corresponds to silent carrier or a proper TAL minor or carrier, and then a HPH disease. So same technique that we saw previously, we use the MLP again. So that will tell you this is how if it's a single gene deletion it will look like. So how many alpha globin genes are there? Yeah, you have alpha 1 and alpha 2. So you have totally 4 alleles, because 2 alleles for each this thing, alpha 1 and alpha 2. So that is why there are 4 possible deletions, single, 2, 3 and 4 deletions. So if it's a single gene deletion, this is how it will look like. Just like we saw earlier, there will be a ratio of 0.5 for a stretch of sequences, and that corresponds to a single gene deletion. If it's a 2 gene deletion, so again we have something called as a cis and a trans. So what do you mean by that? Anybody? Yeah, exactly. Good. So cis is when we have, say here we're talking about alpha 1 and alpha 2. If alpha 1 is normal, but both the alpha 2 are deleted, that is what we call as a cis deletion. Okay? If it's a trans deletion, you'll have one alpha 2 and one alpha 1 normal, one alpha 1 and alpha 2 will be deleted. So this is the difference. So if it's a cis deletion, usually this is how it will look like. You can see that the ratio is 0, meaning in both the alleles, alpha 2 is deleted. So this is another way, another test, which we call as a gene dosage. So again, the concept is the same. This is a normal gene and these are the alpha globin genes. So you can see here, the alpha genes have the same height as the normal gene. Whereas here, if you see, alpha 1 is similar, but alpha 2 is only half of the normal height. So that is a single gene deletion. Here you have only alpha 1, there is no alpha 2. So this is what you just said, it is a cis deletion. Okay, and this is how that corresponding MLPA will look like. Now this is the other form of the trans deletion, where you have a very large deletion which is deleting the entire alpha globin gene cluster. So in one allele, both alpha 2 and alpha 1 are deleted, but in the other allele, the alpha 1 and alpha 2 are both normal. So you'll have a bigger stretch of deletion which is covering both alpha. So this corresponds to alpha 2, this is alpha 1. So both alpha 2 and alpha 1 are deleted, but the ratio is 0.5. So that means you still have one normal alpha 1 and alpha 2. So this is a trans deletion. Okay, and then you have what we call as the HPH disease, where you have this big deletion also and one smaller deletion also, which results in the HPH disease. Okay, so all of you would have heard about alpha deletions, but have you heard about alpha multiplications? Okay, this is something which is equally, if not more important than the deletions. Why? Because this is what is largely misdiagnosed. So what the multiplication, going back to the pathogenesis of thalassemia. So what happens in beta thalassemia? Your beta is mutated, so there is no beta globin chains produced. That is not what is causing the symptoms in the patient. Because there is no beta chains, there is a lot of excess alpha chains which is getting precipitated, right? And this is causing all the symptoms. So what happens if there is alpha multiplication? There will be even more alpha chains which are getting produced. So even if it's a heterozygous beta, because there are excess of alpha getting produced, so heterozygous beta will be a thal minor. So, but now because you have so much of excess alpha, what was supposed to be a thal minor will present as a thal intermediate, or very rarely even a thal major also in some scenarios. So that is why it is very important that you pick up these multiplications, because that it helps to avoid the misdiagnosis. So if you do look for only beta, you get a heterozygous mutation, and you call it. The patient is only a thal minor, but he is recurring transfusions every second month or every third month. Probably that is a good candidate that you look for this multiplication. Just similar to deletion. So deletion, your ratio becomes 0.5 or 0. If it's multiplication, your ratio increases. It goes beyond 1. So here you can see the ratio is 1.5 or even 2, which means there is a multiplication. Okay? So that was just to give you some idea on what we do in thalassemia. In hemophilia, again, for the same reasons, we do the molecular diagnosis. So hemophilia generally, when we talk about mutations, in severe hemophilia at least, hemophilia A, the common mutations is something what we call as intron 22 inversion and then intron 1 inversion. Okay? So these two are detected by specific PCR tests. If they are negative, then we have to look for the whole factor 8 gene. We have to look for the point mutations. Okay? And rarely, if there are no point mutations, we look for deletions also. If it's hemophilia B, directly we go for look for point mutations. In fact, hemophilia B is factor 9, right? So we look for point mutations in factor 9 gene. So earlier we were doing sequencing. Now we are doing factor 8 assays by not factor 8 assay, the factor 8 mutation analysis by NGS. Okay? So then a little bit in the malignant side, hematological malignancies. So why do we need molecular diagnosis? It could be for, identifying the mutations that define the disease itself, like BCR-ABL1 translocation or, JAK2 mutation in the case of polycythemia, etc. It could be purely for the sake of prognostication, like TP53 mutations and many other mutations. Or sometimes it could be for the clinician to make therapeutic decisions. Like common example is your CML, where CML, of course, it will have the BCR-ABL1, but now Even in ALL, you have something called as, it could be pH positive or pH like ALL. Then the clinician can actually decide upon using kinase inhibitors apart from using a chemotherapy in the patient. So it helps to make therapeutic decisions also. So yeah, this I want you all to keenly follow. I mean, it helps for you to understand also. So how to approach a case of leukemia diagnosis. Okay, so again, starts of course with peripheral blood picture morphology. I'm talking only about acute leukemias. Okay, so and then we do a bone marrow aspirate and biopsy. So generally, what happens in our setting is morphology report, of course, takes 2 days at least in CMC. So by then, your flow report will be ready. So by flow, you will get to know whether it's a case of AML or ALL or mixed phenotype or APML, whatever. So once you know the diagnosis, that will decide the subsequent tests. What molecular test you want to do. All that will depend on your flow cytometry assay. So at the time when we are doing an aspirate, we collect samples. One goes for morphology, one for flow cytometry, and one for cytogenetics and all the molecular assays. So when we have the flow diagnosis, we'll decide what fish panel or what karyotype, of course, will be done for all patients. What molecular assays will do will depend on the flow report. So if it's a case of ALL, so we look for specific translocations which have a lot of implications, like 922, 1221. These are all the translocations that define your ALL as per your WHO criteria. And then MLL rearrangements, etc. So all this we were doing by RT-PCR or FISH. Now we have started doing it by NGS also. If it's a case of APML, you look for the classical translocation or all the variant APML translocations. Again, by either karyotype. APML is though the prognosis is very good, it's a medical emergency. You need to have immediate report. So we do something called as a stat FISH, which gets ready within 2 or 3 hours. And but remember, you have to always or rather supplement the one test with the other, because none of the test has 100% sensitivity and specificity. So we do stat FISH for all patients, and in cases where it's negative, you also go back and look at the karyotype, or we do an RT-PCR for the same translocations. More often than not, this could be one of the variant translocations. And then if it's a case of AML, so in ALL, we generally look only for the translocations. AML translocations and point mutations are equally important. The important point mutations, so what are the point mutations you know of in AML? NPM1, FLT3, okay, CEBPA. These are, of course, the defining point mutations. And then, of course, you have the translocations also, 821, inversion 16, etc. Again, by different methods. Now we have NGS, which can identify both point mutations as well as the, all the fusions or the translocations. Okay. So RT-PCR, we generally use it for picking up all the fusions, like BCR-ABL1, for example. Not going to the details, but just to give you an idea how RT-PCR works. So what we do is we have, like any PCR, this will also have two primers, one forward and one reverse primer. Along with it, if it's RT-PCR, that is it. If it's RT-PCR, you also add a probe so that you get the signal that you can identify. So the concept is you have one primer sitting in PCR, you have the other primer sitting in ABL. So what will happen in a normal individual? Nothing will happen, because one is in chromosome 9, one primer is in chromosome 22. There's no way they can amplify, come together and produce a product. But what happens when there is a translocation? Your segment of 9 and 22 are now brought together. So now the two primers, which were on two different chromosomes, they are brought in a close proximity to each other. So now when you do a PCR, these two primers will come together and amplify and produce a product. Okay, this is how an RT-PCR works. So if you get a band or if you get a line like this, that means the fusion has happened or the translocation has happened. If you do not get any line, like in the lane 2 and lane 3, that means there is no translocation in that patient, whatever translocation you're looking at. Now this is the basic concept, which is true for all the, whether you're looking at 922 or you're looking at 821 or 1517. This is how the RT-PCR works. RT-PCR, the same thing, you just add a probe with it so that it's a live. RT-PCR is nothing but, actually it is called as QRT-PCR, quantitative real-time PCR. So quantitative because you have a standard curve at the end of it, and from the standard curve, you get to know the exact copy number also. So it is quantitative, and it's a real-time, meaning as and when the amplification is happening, you can see on the screen all this fancy colors coming up, which will tell you what is happening inside. So different reasons why we do an RQ-PCR. Most commonly, we have, that's how we follow up a patient of CML, by looking at the response. You have the major molecular response, etc. If it's a case of MPN, myeloproliferative neoplasm, what are the mutations that cause MPN? Yeah, JAK2 is the commonest. So we start with looking for JAK2 mutation. If that is negative, then we look for MPL and CALR mutations. Okay, but you have to remember, MPL and CALR generally are seen only in ET and PMF, not in PV. Okay, PV, almost all the cases is going to be JAK2, either most commonly JAK2 V617F. Occasionally you have other JAK2 mutations localized to exon 12. We call it as an exon 12 mutations. So these are the four things we broadly look at. Okay, JAK2 V617F, JAK2 exon 12 mutations, then your CALR mutations and MPL mutations. Okay, this was your, not even the last to last WHO classification, but from the last classification, we have something else apart from this. What is it? We call it as a triple negative myeloproliferative neoplasm. Meaning, triple negative meaning these three hotspot mutations. Your V617F, MPL and CALR mutations are negative, but still the morphology is very clear-cut, pointing to a picture of myeloproliferative neoplasm. Okay, again, more with respect to ET and PMF. So this is what we call as a triple negative. So those cases we do a next generation sequencing. So more often than not, they will have some other mutations, like SF3 beta 1, ASXL1, TET2, etc. You don't have to remember all this, but you have to know that there is an entity called as a triple negative MPL. So yeah, this is the same thing. So if it's PV, we don't generally look for CALR or MPL, because it's almost always only JAK2 mutations. So we do that. Then there are other rare scenarios like clonal eosinophilic disorders, where we look for all the PDGFRA, PDGFRB fusions, etc. You can see over here. Okay. So these are some of the common ones. Then there are a lot of other germline and somatic disorders, like I've listed a few over here. So earlier we were doing all this by different sequencing assays, which extremely painstakingly laborious, and now they're all simplified to a great extent because we have next generation sequencing. So you've all heard about NGS? Yes, I mean, anybody and everybody talks about NGS these days, but what exactly is NGS? Any idea? Okay, so yeah, you don't have to know the technical part of it. I mean, understanding the whole concept of NGS is, it's very difficult. Even I'm not fully aware of everything that goes into NGS, especially the bioinformatics part. So as a pathologist, what you should know is, when to do NGS to begin with. It's not like everything and anything. You get a sample, get a patient, take a blood, send for NGS. That is not the way forward to go. And why do we need NGS in a diagnostic lab? I've shown so many other techniques that we have been seeing so far. All the PCRs, your MLPA, all your RT-PCR, RQ-PCR, etc. So where exactly does an NGS fit in a diagnostic lab? And when does a pathologist need to do a test for NGS? So I'll just give you a few basic scenarios where NGS is really helpful and where we should do the test by NGS. Okay. The first example, what is the disease in the patient, what is the gene mutated, but the gene is so big that it's very difficult to sequence it by convention. You have to remember, all PCRs and sequencing can only amplify up to say 1,000 bases. So if you're looking to look for mutations in a gene which is like 100,000 bases or something, you have to do so many 50 to 100 PCR reactions followed by 50 to 100 sequencing reactions. That is, it's not. One sample might take so much time to get the final result. Classical example is factor VIII. So, the factor VIII, if you see, it has 26 exons, and you can see one of the exons is actually very big. So when we were doing sequencing, of course, we were using different other techniques for this. But if we were to do a sequencing, we'll have to set up 35 PCR reactions for one sample. So you can see the amount of time and the cost that it is going to take. This is just one example of factor VIII. There are so many genes which are extremely, in fact, most of the genes associated with solid tumors are all very big. So then this is where your NGS will help. You can, just take the sample. You don't Unlike a PCR or a sequencing where you have to set up individual reactions and do your NGS, you just take the sample, put it in the NGS. Not just one factor A gene, it can even get the whole genome sequences from that sample, taking the same amount of time. So it really makes a difference when you're looking at mutations in gene which are very big. Okay. The next example is when you're still very clear what your diagnosis is, but when you have to do a molecular diagnosis, there are so many genes that can be mutated, causing that particular disease. Okay. One example is over here. So what are you seeing here in this? I'm sure you all will be better morphologists than I am right now. So, yeah, this is a very typical picture of a hemophagocytosis, right? And a patient with this picture, pancytopenia, hyperferritinemia, hypertriglyceridemia, hypofibrinogenemia is a very typical presentation of HLH. So you know the diagnosis for sure. Let's say this patient wants to go for family genetic counseling or something like that, in which case you need to identify the mutation. The problem is HLH can be caused by mutations in nearly 15 different genes. Although 3 or 4 of the genes are more common, still those 4 genes and some of those genes are very big. So again, the same problem, having sequencing so many genes and setting up so many reactions is going to be very difficult. So this is where again your NGS is actually going to help you. The third example is when the patient phenotype is not very clear. What we have seen fitting very clearly into this entity is primary immunodeficiencies. Currently there are 485 disorders grouped into this broad waste basket called as PID. So some patients will have very clear phenotype, but not everybody is going to have typical features of that particular PID. So all you can say is that maybe some inborn error of immunity, but you're not able to characterize it further. So these are the areas where NGS really helps. When you do an NGS, you will get to know exactly what mutation you have, and based on that mutation, what particular disease spectrum you are looking at. Okay? And then, of course, all your solid tumors and all the hematological malignancies, where you need to look at a panel of genes for a clear-cut prognostication in the patient. And these are some of the genes we look at in a myeloid malignancy, MDS or AML, etc. Don't get scared by all these fancy pictures. This is nothing but just to show you that there are so many possible. So when we say ALL, we think of 2-3 translocations. When we say AML, we think of maybe 821 or 1517 or inversion 16. But there are hundreds of possible translocations that can result in that mutation, which earlier we were not able to identify, but now we can because we have NGS. Just to highlight you the importance that NGS can make, we now have. You have heard about PH like ALL? Okay, so the current classification, earlier classification, it was a provisional entity, but now we have a definitive entity called PH like ALL, meaning it's very similar to BCR-ABL positive ALL, but it doesn't have the BCR-ABL1 translocation. Okay, this whole PH like ALL concept came about when NGS came into the picture. By doing RNA-seq and DNA-seq simultaneously, they were able to identify a similar expression profile to a PH positive ALL, but the mutation was absent, which was grouped as a PH like ALL. And to tell you how much of a difference NGS makes, so you've heard about the Human Genome Project. So this was an attempt started, I think, 1990, where the scientists from multiple institutes wanted to get the entire human genome sequenced. They started in 1990. This was by a team of nearly 100 odd scientists coming together. And one sample, by the time they got the whole genome done, it took them 13 years to get the sequence. They started in 1990 and finally they finished it in 2003, sequencing by nearly millions of PCR reactions, then merging all those data together. By the time they finished this is when NGS came into the picture. And shortly after they put this data of 13 years for one patient, they had NGS and they were able to do it in 2 days. One person was able to do it in 2 days. That's how much a difference NGS makes with respect to the earlier technique. So it's like comparing to your first computer that you had versus your current smartphones. The whole genome project actually cost them $1 billion US, and took them 13 years. Now it's out. In fact, you can get it done at $500. You can see the difference, and the current machines you can get it in 6 hours actually. So that's how much difference your NGS does. So I'm not going to talk about NGS for you, just to give you some idea what happens. Again, like any PCR, you start with DNA. Then, depending on what panel you are trying to test, next step is to prepare that panel, what we call as a library preparation, and then you take it to the sequencer. And then there's a whole lot of computational bioinformatics that goes into it. I'm not going to tell you all that. And at the end of it, you will get an output which will tell you basically everything you need to know. It will tell you what gene is mutated, what particular mutation it is present. It will tell you whether it's a polymorphism or a mutation. It will tell you what is the disease associated with that particular mutation. It will tell you whether it's a pathogenic or it's not a pathogenic mutation. So anything and everything you need to know, you will get the result at the end of all the analysis from the NGS. So maybe that will be too much for you right now, but for you to understand, basically NGS has a sequencing part and the bioinformatics part, and at the end of both, you get the final output. Okay. There are different ways by which you can use the NGS. Where you can sequence the whole entire human genome, or only the coding regions, which we call as an exome sequencing, or you can look at specific panels, like say for example, a case of AML. So we have specific panel for myeloid malignancies, which will have only some 60 or 70 genes. So rather than sequencing the whole 21,000 genes that you have, you can only sequence these many number of genes and then do the analysis. And there are, if these are all done with DNA as a starting material, then there are scenarios where we use RNA sequencing also. I'm not going into those details. So, for you to easily understand, I'll just give you two crude analogies. What exactly is NGS? It's something like performing a surgery. Okay? So in surgery, you need a surgeon and you need an anesthetist. Only when the anesthetist does his part, the surgeon can actually go and operate. So that is what an NGS is. So you need a bioinformatician and you need a pathologist or a biologist. Okay. So only when the bioinformatician has done his part, has done all the, run it through the pipeline, and you have the final output. Then comes the role of we call ourselves as genome analyst, essentially the role of a pathologist, where you will have to look into all those variants that you have and identify which is the one pathogenic variant which is causing the mutation. Okay. And again, another crude analogy for you to, if I have to explain you what I do with the NGS, so this will tell you. Basically what the NGS machine does is, it throws at you a lot of garbage, okay? Tons and tons of garbage. You have to dig into the garbage and you have to come out with one mutation which is going to explain the disease in the patient. So what does this mean? So this is what happens. So at the end of the bioinformatics, this is from a exome sequencing. Okay, so we have somewhere around a data of almost 5 to 6 GB generated from the single sample if you do an exome sequencing. And once we run it through the bioinformatic pipeline, we get around 90,000 variants. Hits. Each SNV will be shown as a hit. So we'll get somewhere close to 90,000 hits. So what we have to do is we have to apply various filtering criteria based on multiple parameters. I'm not going into those details. And eventually come down to that single variant which is going to explain the disease in the patient. So basically, this is what the NGS analysis means. And there are different areas where it has made a huge, impact. If someone you had attended yesterday, this was one of the posters there from one of our fellows on how it has changed our approach to platelet disorders. Like earlier, when we think of platelet disorders, we'll say probably, of course, Viscott aldridge, but if there is something abnormal in the thromboelastograph, for example, we'll think it might be a case of Glanzmann thrombasthenia or maybe Bernard-Soulier syndrome. But you can see there are so many. Within just 1.5 years, we have identified so many other platelet disorders, and not just there were of course case of Glanzmann thrombasthenia and BSS, but there are so many other disorders which we were not picking up earlier. But now, because we are doing an NGS, we are able to actually diagnose all these patients. There are different other areas where we use it at hemolytic anemias other than thalassemias, enzymopathies, all your membranopathies, etc. And it helps you to understand a lot of details. And this was one recent example where it's a case of PID. With the typical clinical history, normal B cell and T cell count. So clinically, we were not able to diagnose what it is. We did an NGS and we picked up a mutation in one particular gene, DNMT3B gene. Not going into the details. It also teaches us a lot more about the pathogenesis and the disease we are looking at. Like this was the first case of
DNMT3B that I saw. And when I read about it, I realized that there is something very fancy in a karyotype in this particular gene mutation. And so we did a karyogram, and we got a very beautiful, something like an octopus kind of a picture. So it teaches us a lot about what happens in the disease biology. Ultimately, it all comes down to identifying the mutation, and NGS really helps in that particular scenario. So yeah, before I think probably a lot of you would have slept by now. So before I conclude, just wanted to drive home the idea is, like I said in the first slide, when I was sitting there, I thought, morphology is the whole world, but there is so much more beyond morphology. I'm not saying morphology is no longer redundant. Of course, its significance has come down in hemat especially. And some might scold me if I say that, but yeah, the importance of morphology as every year goes by, morphology will become less and less important. It's all these high-end techniques that is going to come into the picture. So it's important that whether you're moving into a career in hemat or solid tumors or even just general histo or whatever it is, molecular diagnosis. When I take molecular, I'm including both cytogenomics, cytogenetics and molecular changes. All these are very important, and the field will keep evolving. So when I joined CMC, which was not very long ago, I left KGMC 2015. So during my 2-year training, I was learning all about Sanger sequencing, RT-PCR, real-time PCR. After I became a faculty, all those tests were gone. Within a span of 3 or 4 years, whatever we were testing by Sanger sequencing, we've started doing it by NGS. Whatever we were testing by RT-PCR or RQ-PCR, we still do it, but a lot of it has been replaced by something like RNA sequencing or droplet ddPCR, etc., etc. And everything keeps changing. So the field will keep evolving, and you have to keep yourself updated. And that's what I'm trying to say. And with that, I'll stop. Of course, it's not in full screen now, but yeah, I cannot stop without thanking the person who is responsible for me standing over here. I don't know if sir was here when you all were students, but I came to Lucknow from a very different place, as a total stranger coming from a completely different cultural background and everything. I needed somebody to hold my hand and guide me, and that was the major factor that pushed me. And he's the one who is fully responsible for it, not just for my MD. Even after MD, 3 years I got very fond of Lucknow, so I wanted to stay back. I was getting SRship in SGPGI. I went and told him probably I should do this. And one more important thing is CMC doesn't pay you that much. So I told him probably SGPGI is better for me. He said, just shut up. Even if CMC doesn't pay you a single rupee, you go there. Trust me, that will take your career to a different path. And if not for those words, probably my career would have been in a different way. So all credit to him, and good luck to all of you, wherever you do.
Thank you so much, Arun, for taking out time and coming here. And it's always a matter of happiness and pride when we see our students achieving the newer heights, a greater height in their lives. So we wish you all the best. And when Arun joined here, he was a very silent student. And he used to work silently, and like whatever work we used to give him, he used to do that. And God bless
you. I would consider myself silent now also.
Yes, of course, you can ask questions like, if any of you have but what Arun said is very true. This molecular pathology is the future of pathology. Morphology is good. Of course, morphology is important and nothing can replace morphology. But as the time passes, this molecular pathology is going to take up. So you must do fellowship, or you must do some sort of training in the molecular pathology. It is must now.
Especially in hematology.
In hematology.
Classification is based on.
This is the future of hematology now. The whole classification is based on molecular and cytogenetics.
So you don't have to need like अपनी बोल रहे ना कि ancillary या extracurricular type में थोड़ा सा knowledge दिख लिया, but now it should become a major part of it.
So if they are very
Actually, it's not something very different. I mean, I've never seen it. I mean, I've seen PCR very, but I've never worked with PCR till I finished my MD. So only when I joined my fellowship, I got exposed to PCR, and I was thinking this is something. Till then, whatever when I say molecular techniques, mutations, blah blah blah, these are all something very fancy. What exactly is it? I was not able to make an idea out of it. But once you get into it, initially it will seem very complex and complicated, but give you a few months' time, and it's not that complex a thing to understand. So, as long as you are in a facility where you have exposure to all these techniques, it's relatively easy that you can learn all this.
And one more thing, you should be ready to accept the changes. Like what he did, he left Lucknow, he went outside. You should leave that comfort zone. You should be ready to learn new things and accept the new challenges. That is very important.
That is true. If you want to learn something, you will have to go there to learn. It is not going to come to you.
Leave the comfort zone.
वो comfort zone से तो निकलना पड़ता है, ये सीखना।
Take the timely decision, leave the comfort zone, go and learn newer things, achieve newer heights. All the best to all of you. Thank you so much, Arun. Thank you.
I'll be here for some more things. As ma'am said that if you have some questions, what to do after MD, you can ask.
You can ask Dr. Arun. We will share his number if you permit on the
He is there.
He's there. Okay.
So when you will get in contact, but from my line will be in the left line. And everyone has to share कि वो कहाँ employee हो रहे हैं. Because we have to keep a record.
On behalf of HOD, sir, we invite all the faculty for ID.
Thank you, Doctor. Very nice, excellent. अब आप थोड़ी देर स्टूडेंट्स के साथ रहिए, हम लोग चले जा रहे हैं, और
Maybe they're all a little skeptical to ask because faculties are
हम लोग ये भी